Today we're going to be talking about biomodulators, and this will be largely covering the material in your Janeway textbook in Chapter 16, Sections 1 through 12. And I'll be showing some figures from the textbook, and the figure numbers will be on those slides, so you can refer back to the textbook. So today we're going to be talking about, just starting with, what are biomodulators? And then we're going to be discussing different types of modulators. And really, it's not important that you guys remember the names of all these different drugs and medications. What's really important is that you understand when you might want to use each drug, so why these drugs are given, and how you might choose the correct drug based on balancing the immune effects as well as possible side effects. So some of the drugs we'll be discussing first are corticosteroids, then cytotoxic drugs, cyclosporins, and finally we'll talk the most about monoclonal antibody. And we'll be discussing this because this is really a cutting-edge area in medicine where we can use highly specific monoclonal antibodies to target exactly the part of the immune system that we're interested in. Okay, so to start out, what are biomodulators? So biomodulators that we'll be discussing today are used for treatments for unwanted immune responses. And typically this would be something along the lines of autoimmunity, organ transplant, or allergy, where the person is having too strong of an immune response and you want to give a medication to help dampen that response. So the goal of biomodulator drugs are to help avoid tissue damage from an immune response and also prevent disruption of the function of different tissues. And as I mentioned in the last slide, these drugs can be different small molecules and also now the new category called biologics, which include monoclonal antibody therapy. And one thing to keep in mind as we go through this is that all of these drugs, no matter how specific they are, will target both helpful and harmful immune functions. So I really want you guys to keep that in mind. So we might be dampening the immune response, but then, for example, that could leave someone open to infection. Okay, so this figure 16.2 is just a summary of some of the different drugs we're going to be talking about. And we're really going to be focusing on the corticosteroids, the cyclosporins, including rapamycin. And monoclonal antibodies, which are not on this list. So starting out, corticosteroids are a really commonly used medication that is very broadly anti-inflammatory. Corticosteroids are derivatives of the glucocorticoid family of steroid hormones, and the most well-known is a medication called prednisone. So this is essentially synthetic cortisol. And because these are hormones, they act similarly to natural hormones in the body. So they'll pass through cell membranes and bind to receptors on the nucleus. So corticosteroids, we say, they're broad anti-inflammatory drugs because they actually can change 20% of all the genes that are expressed in leukocytes. And some example targets for these drugs would be to dampen pro-inflammatory functions of monocytes and macrophages, to decrease the number of CD4 T cells, and to decrease overall cytokine production. And it's really important that you guys consider some of the negative effects, because while this might help with a broadly inflammatory condition, at the same time, these functions of the immune system are also essential for normal function. So, when might you want to use a corticosteroid? So again, really, this would be your general first-line therapy when broad effects are wanted. So if you have a disease that's causing inflammation throughout the body, this is a quick way to dampen a lot of that. It's also used commonly in diseases such as autoimmune diseases, where the mechanism of the disease is not known. And in cases of these diseases, they might not be well targeted by other drugs. So giving this very broad therapy is a way to try and reduce some of the symptoms the patient is having without having to know exactly what is affecting them. And as I mentioned, it's a really first-line therapy, and people tend to try and keep this to a short term because it has quite broad side effects as well, as you might assume from the broad function that we see. So a few examples of uses for this would be an initial therapy in an autoimmune disease, and this is coming less common with monoclonal antibody therapy. We might also see it in something like a severe allergy, for example, a skin allergy to poison oak or another plant that causes a rash. And this is also sometimes used to break a cycle of inflammation, for example, in migraine headaches. So if someone is having a cluster of migraines, they just can't get those migraines to stop, this is actually used as a therapy to help dampen overall inflammation and try to get those under control. So here's a table that really is summarizing the broad functions of corticosteroids. So you can see all these different cytokines that this medication has an effect on, and this is going to broadly dampen inflammation. We also have a reduction in nitric oxide production, as well as other essential molecules for immune function, such as adhesion molecules, which, as you guys have talked about previously, are very important for leukocytes entering tissues to fight an infection. So another category of drugs are cytotoxic drugs. And these are really also quite broad in their mechanism, and they cause immunosuppression by killing dividing cells. So why does this target the immune system if this is so broad? Well, this interferes with general DNA synthesis, and so it largely acts on rapidly dividing cells. And one of the cell types that is most commonly rapidly dividing are hematopoietic stem cells in the bone marrow. So, as you might guess, however, this drug class can have severe side effects since it is essentially acting on every cell in the body. So these drugs are typically only used in patients who are going to be receiving a bone marrow transplant. And in those patients, you want to kill off the hematopoietic stem cells that are in their current bone marrow, so that will leave room for the transplant. So then we reach another category of drugs, and these drugs inhibit lymphocyte signaling. So a few examples, and we'll talk about these and their specific inhibitory effects, include cyclosporin and rapamycin and JAK inhibitors, for a few examples. So these drugs really help stop clonal expansion of lymphocytes. And cyclosporin and tacrolimus inhibit calcineurin and NFAT in order to achieve this. So this really helps inhibit signaling and therefore the production of cytokines like IL-2. And this generally will inhibit T cells as well. And it's used to prevent transplant rejection. So after a person receives a bone marrow transplant, they can be given these drugs to help to prevent that transplant from being rejected. And again, you really have to keep in mind the potential negative side effects that these drugs might have. Since they still act fairly broadly on all the T cells, sometimes B cells, you want to make sure that you aren't completely inhibiting that. So we want to try and stop transplant rejection while also minimizing the side effects. So here in figure 16.4, we have some examples of how these drugs affect signaling in T lymphocytes, B lymphocytes, and granulocytes. So here you guys can review this in your book, but you see all of the cytokines that this affects. So we see reduced expression of IL-2, IL-3, IL-4, GM-CSF, which is an important growth factor, and TNF-alpha. We also see an inhibition generally of proliferation of these cells. So here's a more specific signaling pathway showing how these drugs act on NFAT. So calmodulin and NFAT are really important for transcription of certain cytokines. In this example, about IL-2. So here we can see that in the normal pathway, NFAT, after calcium signaling occurs, is released so it can transfer to the nucleus and allow transcription of genes such as IL-2. In contrast, if we have a drug binding, in this case cyclosporin A, this is blocked. So NFAT cannot be activated and therefore it cannot transfer to the nucleus and transcription will be blocked. Okay, so the next couple drugs in this category are rapamycin and JAK inhibitors, and these also block signaling. So rapamycin specifically blocks the mTOR pathway, which is really important for growth and proliferation of cells. It also, because of this, limits effector T cells and increases regulatory T cells. The JAK inhibitors, or JAK inhibs, block JAK signaling, and this is often a signaling pathway that is downstream of cytokine receptors. So here we can see an example of rapamycin acting. So it disrupts these two branches of the mTOR pathway, which are important for things such as the actin cytoskeleton and cell proliferation. And you can see, particularly in the signaling pathway, where this is blocked. So the next and largest section of this lecture will be talking about monoclonal antibody treatment. So these, as you might guess, are much more specific and typically have fewer toxic side effects. And that's because they are specifically targeting certain proteins or cell types that are important for this disease. So there are fewer off-target broad effects. So one type of antibody treatment is called IVIG, or intravenous immunoglobulin therapy. And this is just a variety, many immunoglobulins that are infused into a patient. And this can typically help boost their immune system. We've talked about this as well in the immunodeficiency lecture. So one thing that you have to be careful of when using monoclonal antibodies is the possibility of serum sickness. So serum sickness occurs when immune complexes form, and this is typically if an animal immunoglobulin is given. So the patient, the human that receives this treatment, will mount an immune response actually against the foreign antibodies that were infused. So now we'll talk about About a couple different types of monoclonal antibody treatment. So the two main types include depleting antibodies. So this means that these antibodies bind to cells and target them for destruction by macrophages and NK cells. The other type is a non-depleting antibody. So here, we're not getting rid of these cells. We're actually blocking the function of target proteins. So I mentioned serum sickness earlier. So how can we reduce the chances of that happening? So it's much easier to make an antibody by immunizing a non-human source. However, this can trigger immune response and also allergy. So humanization is the process of making antibodies that are not recognized as foreign by the immune system. So there are a few different categories of humanization. So the first is a chimeric antibody, and this is typically designated by the suffix iximab. So an example of this is rituximab. And in chimeric antibodies, the antigen recognizing region from an animal might be spliced into a human Fc region. So the area that's being recognized by other parts of the immune system is seen as human. So we can go a step further, and actually, instead of replacing the whole antigen-presenting region, we can just replace the hypervariable regions. So if you guys think back to previous lectures where we've talked about antibodies, you'll know that these are really important for antigen binding. And again, these have their own suffix. And this, you can always tell if something's a humanized antibody by Zumab. So finally then, we have fully human antibodies. So these have no areas that are derived from an animal. They're fully human, and they're designated with UMAB.